and results, and also related challenges for different stem cell-based system. So now it is neural stem cells. And so this is a second system, stem cell system, which we have been working intensively on since some years. But it's also interesting to see to look at this, because it shows that in fact different systems, even if we are interested in similar type of questions like cellular hierarchies and so on, so then they pose different challenges, both on mathematical level, but also on the level of data acquisition and understanding of the data. So now something is not working with my file. I just share again. So wait, sorry. Okay, good. So neurogenesis. So we are interested in stem cell dynamics, which produce neurons, and they are located in the brain, and there might be several locations now, depending on the system which we are looking at and on the applications. And for certain reasons, in this system, we cannot look at the human data. So here we are interested in understanding of the data coming from adult neurogenesis in mice. So this is in collaboration with the laboratory of Anna Martin Villalba at the German Cancer Research Center in Heidelberg, but also some other collaborations, which I will mention later. And so here we can look at two places when the stem cells are located. One is subventricular zone and one is hippocampus. And in what I present today, we mostly look at the subventricular zone. So these are neural stem cells which produce olfactoric cells. But concerning this, the structure of the system, it is as we had it in blood, that we have a population of stem cells which then divide. And then after division, they produce more stem cells, or they differentiate and produce some progenitors, and then finally differentiate into neurons. And again, at this system, we can look at least for a set of questions we have without looking at the spatial structure of the niche. This is also that finally neurons are not staying there, but just moving from the place. And the second observation is that in fact the processes look spatially statistically homogeneous. So now, as I'm always saying, the way how we approach the problem depends on the questions and on the data. And first, I would like to start with modeling in case of more classical data. So here, these are data related to cell numbers, and the cells which can be measured are stem cells, but also like progenitor cells. In case of neurogenesis, they call it TAPs, from transmit amplifying progenitors. And the problem of neurogenesis is that now you have to kill mice to get data. So certainly, if we need a time course, we don't get the data from the same mice, but it is assumed that the mice are, because they are genetically the same, so it is assumed that we can take a different data in different time points from different mice, and that it will reproduce the representative dynamics. And here, if you look at the data, you see the black dots, and the black dots come from different mice that were used to get measurement for the same data. So in fact, we see that there is some heterogeneity among individuals. Okay, so then we have the data in 3 time points, like young, 2 months old, 7 months old, and then old, which are 22 months old mice. And what we see here, and what is a big difference to this, what we saw in blood and what we are interested in blood, is that here you don't have a stationary distribution in time, that in fact in healthy system you observe strong decline during aging. And the question is, how can we explain this behavior? So if we just think about stem cells, so the difference also to all our blood considerations from Tuesday is that now they are measuring somehow separately 2 types of stem cells. One are quiescent stem cells and then active stem cells. And quiescent stem cells are these which are just sitting there, and then they get activated, and then these active stem cells are stem cells which are cycling. And then they divide, and after division, as always, we get 2 daughter cells. And depending now if division is symmetric, asymmetric, and so on, so the same as in blood, with some you get some fraction of cells which stay stem cells. So in this case, they land in the compartment of quiescent cells, and 1 minus this fraction which differentiate. So now our stem cell population is described using 2 equations, and this is the new aspect comparing to the blood models we had. So these are the quiescent cells and active cells, but then the logic of the system is, as I exactly said, that quiescent cells get activated. So we have important parameter A, parameter of activation, and from this we get active cells, and then division at the rate P, and we get 2 daughter cells, and with probability B it is stem cell, and 1 minus B is differentiating. So if we just look at the stem cell equations, we have these 2 equations, and of course the same as in blood, we can extend it to take care of the rest of the lineage, especially if we would have this measurements, and we have these measurements for progenitor cells, and sometimes also for neuroblasts. So now, however, since at the very beginning we don't know about regulations, and we also observe this decline, we ask ourselves, could we at all maybe even explain this decline process with just linear equations? Maybe maybe parameters are constant in time. So if I don't have feedbacks from the downstream compartments, I can look at these 2 equations just independently, yes, because they are only coupled through this transition and through the proliferation. So then it is really the simplest possible model one can imagine. We have just 2 ODEs, and even at the beginning linear ODEs. And this is interesting that one can help understanding biology even with so simple models. And then because in the first step, what we immediately see is that the decline which you observe in data, now you see this decline here in a logarithmic scale. So this decline is slower than it would be if it would be exponential decline. So red line is solution of this linear model fitted to the data, and of course we cannot fit this data. There is one more aspect which is here now important, is that we have also measurements of a fraction of active cells among all stem cells. So it means here you have data for A over A plus Q. These are these black dots. So if the model would be, or if the process would have constant parameters, so if we could use this linear model, then this quotient, this distribution would very quickly converge to a steady state. So it's like with the red lines, you would have here constant, and we observe here decline. So this already indicates that there is a regulation of the process. So the process is not homogeneous in time. And now the question is how the process is regulated. And again, before guessing about nonlinear feedbacks, as we did in the blood case, we can simply ask the question, which parameter must be regulated in time to describe dynamics which we observe? And we have just 3 parameters, R, B, and P. So the question is, which parameter is, let's say, changing so that it looks like the system tries to, or the system takes care of not losing stem cells too fast, yes? So one idea would be proliferation. It's always the most intuitive, and one now also could say, if I don't want to lose stem cells too fast, I just slow down their division. But such model with regulated proliferation would have very similar dynamics to the linear model. And now this maybe it's also important to say it explicitly, that in this very first step we just try regulation of not really regulation of parameter, but changes of parameters just given by time-dependent functions directly. So just first trying to understand which parameter is changing and in which way in time during aging, so that we can then later build hypotheses on the nonlinearities. So the other idea would be that activation. So I want to save my stem cells. So with time, the stem cells are getting lazy, and it's more difficult to activate them. And this regulation seems to work pretty well. So we not only describe this decline in the saturation in the decline of cell numbers, but also we describe this decline of the fractions. So now you have also the third parameter. This is the self-renewal. So now we then can assume that self-renewal is increasing with aging. So it means with aging, there are more symmetric self-renewing divisions, just to, again, protect stem cell pool from too fast decline. And this regulation also seems to describe the saturation in decline and some decay, but smaller. So now the question is, of course, we can now just mostly debate between these 2 regulations. If you look from the purely numerical perspective, just optimizing the error and also looking at the Kaike criterion, then the regulation of activation rate is better. But just to try to understand it even better, we proceeded in the following way. So if I have only regulation of self-renewal, I have this picture, only regulation of activation, this picture. Then the question is, what happens if I regulate both? And then, of course, even a little bit better because I have more freedom in parameters, but in fact, it's pretty similar to the regulation of activation. And now if you look how parameters change, if we allow both parameters to change in time, then the best fit looks like this. Self-renewal is actually constant, nearly constant, and activation rate changes nearly in the same way as if we allow only to regulate activation rate. So the conclusion is that, in fact, this regulation of activation is the essential thing, and regulation of self-renewal, at least to explain this data, could be even neglected. So this was the message and the outcome, and this was new because before experimentalists expected that self-renewal is important parameter, or maybe proliferation, but not activation. So the conclusion is that with aging, stem cells become more quiescent. So now the question is, what is making them more quiescent? So we can compare their behavior looking at young and old cells, and then you can look at the different properties of stem cells, like, for example, their ability to build clones, like how big are clones, how they how much daughter cells you get from the same mother cell, and then the behavior between young and old does not, there is no difference. The same concerning division rounds, they are very similar, and the same concerning length of the cell cycle. So indeed, experiments confirm that this is not proliferation which should be regulated. So now the question is what else we can look at. So one could look at this now distribution of cells with in this analysis of single cell data with respect to different transcriptomes, and especially if you look at this UMAP picture, which shows distribution of different cells and their clustering into neuroblasts, quiescent cells, active, and so on. Then if young are green and the old are this violet, and then you see that in fact they are mixed here. However, if you now look at their concentration or distribution along the pseudotime, you see that indeed in the old mice, there is somehow bigger proportion of cells sitting in the quiescence. So this also is in line with our model prediction. And what one can also do, so somehow like this is example of model-based experimental design, is that we performed comparison of the young and all mice data with respect to gene transcription, and the computational analysis pointed to the changes in signaling, so-called Wnt signaling. I don't want now to go to this molecular details here, but somehow just to tell you that we could this way find out some differences at the level of signaling molecules. This was, in this case, it was just finding that in old there is a bigger transcription of an inhibitor of this important gene, and this way somehow one could come to the conclusion that Wnt activity promotes quiescence, what is also in line with other model predictions. So always, if you want to see how model responses to such hypothesis is good, if one can do some perturbation experiments and then model them. In this case, it was like inducing more of inhibitor of this Wnt, and then producing data. And when model was fitted to this data, again, model suggested that quiescence increased. So I really don't want to go here to details because this Wnt signaling is pretty complicated and still not well understood, but just wanted to show you that even such simple models can be used to guide experiments which are on a deeper level. Okay, next question is does it have anything to do with inflammatory signals? Because what is clear that with aging, a level of inflammation increases. So if we want to understand now the role of this inflammatory signal, it would be also a good idea to be able to perturb it. So here the idea was just to perform inhibition of some receptors for interferon signals. So these are things related to inflammation. And then, so the idea was, okay, we switch off ability of the stem cells to respond to this. So, okay, so far what I plotted here on the slide is this wild type dynamics, what we had before. So we have our model, and we have data from this healthy system. Data are for stem cells and for fractions. And then we have our blue fits. And now what we do, we inhibit these interferon receptors. And then what happens? So what one thought, okay, we inhibit this inflammatory response. So maybe this means we would inhibit this effects of decreasing activation rates. So then one would expect that in fact now we should have faster decline of stem cells. And then these blue dots are now new data. And then, surprise, surprise, okay, at the beginning even it seems that the decline is faster, but for longer times it saturates again at similar level. While the difference is visible in the fractions, because now fractions stay constant. Okay, one can even argue if they not increase a little bit, but this is, let's say, one of the open questions now with discussion with experimentalists. So now our expectation was that with this interferon knockout, we switched regulation of R parameter. We have new data, and now we fit our model when we allow regulation of both R and B to this new data. And now, even more surprise, so okay, blue now were parameters activation and self-renewal for wild type, and red are parameters for the new system, for the system quantified with this perturbation data. And indeed, the model predicted that now activation rate is constant. But if it's constant, then what happened that we have again this saturation? Okay, the model predicts that now self-renewal is changing. So somehow the system compensates. We disabled changes of activation rate, and the system compensates by regulation of self-renewal. But again, this suggests that somehow the primary parameter which is regulated by this in the system is activation, and then self-renewal has a kind of secondary role. Okay, so if we have this model, non-autonomous model with time-dependent parameters, we would like to understand how these parameters are regulated by the system. Can we do something about this? What we had estimated for wild type is how these parameters change in time. So maybe we can plot these functions as a functions of cell numbers. And then we see the shape. We see that activation rate is increasing with increasing number of cell. That's why it is decreasing in time, because with time we have a decline of cells, while self-renewal is decreasing. So question is, can we translate it to cellular feedbacks? And this we can kind of guess that these functions maybe could be described by Hill functions when activation is described by this function where cells promote activation but also inhibit activation, and self-renewal should be just function like self-renewal in our blood models, where we have just decline with the increased number of cells. And we wrote here cells intentionally because in fact at the first glance I don't know which cells. I can say NSC counts, neural stem cell counts, but which neural stem cells? Quiescent, active, or maybe, as in case of blood, we would like to have this dependence on the downstream compartments on the more mature cells. So can we verify it? And then there are two ideas how one could approach it. One idea is to maybe extract more information from these nonlinearities on possible signaling molecules, and then try to link it to all this Wnt and other molecular signaling. So sounds promising, but very difficult for two reasons. First of all, okay, as we did it before for blood models, but in the other way around, if you may remember, if you think about the equations for signals, and then you put the signals to quasi-steady state, you may obtain the nonlinearities. So here we may go another direction, and then you see that the signal which is described by this Hill nonlinearity is in fact always produced by some external cells, like by the niche, and then you have this decline, and then you have this consumption. So in this this case, it would be just binding to receptors of the cell type, okay? While if you have this kind of nonlinearity, you have also this binding and decline, but the production comes from the cells. But of course, one can say, okay, this would be like this if we had here just one signal regulating this nonlinearity. One can also think that maybe I have two different signals. One is just activating and one is inhibiting. So it already shows that there is still some freedom behind. The only what is clear is that if we don't have this type of nonlinearities, for example, if I would like to exchange them, the system could not describe the data. And it's already here. One can also try to link this to some molecular knowledge, just asking which signals are produced really by stem cells, which signals come from the niche. So this is now one of the part of our ongoing work, but again, it is pretty difficult because not so much is really understood on the level of molecular interactions. Okay, so here it's somehow also idea how experimentalists should look like at the system when looking for the molecular signaling, but somehow we cannot really solve the problem which cells are producing which signals and how. So the question is, could we then get anything by looking at specific models? And here we look at three hypothetical scenarios. In one case, we say activation is induced by quiescent cells themselves. So the more quiescent cells I have, the more are activated. And also there is some saturation in this process, while self-renewal is regulated by active cells. There is some evidence in other systems, like in some fish brain neurogenesis and so on. So one can think about such regulation. Another scenario would be to say, okay, activation is induced by quiescence, but then active inhibited, and also active inhibit self-renewal. And finally, one could also, let's say, follow more our ideas from blood system and say maybe even self-renewal is inhibited by downstream compartments. So here there would be neuroblasts. So something what is produced later on by this differentiation process. Okay, so if I had one of this, I could just check if it fits to data. But if I came with three ideas, I have to compare them. And this is very interesting because in fact, the three models behave in a pretty similar way. You can do some analysis, like, it's like only two nonlinear ODEs, so we can do phase plane analysis. And we find something what is very consistent also with our result for blood, namely that if self-renewal is smaller than 1/2, okay, here it would be just this maximal self-renewal, then we get just only one steady state, which is trivial steady state. And if it's larger, then we get a second steady state. However, in this system, on the contrary to blood, for us, it's still interesting to have this situation with only trivial steady state, because we observe always decline in time. So somehow, if you look at the data, maybe I show it here. If you look at the data and you see this decline, you cannot exclude that in fact it's declined to 0 for very large times, or it could be maybe declined to some constant, and then we have this scenario just with such parameters that the second steady state is small. On the level of the data we have, we cannot really decide if this is this or this scenario. So again, when you estimate parameters for the data, usually you estimate parameters corresponding, it seems that this corresponds to this case. So we cannot distinguish using model analysis. Can we distinguish using inference, using parameter estimation? So we can take all data we have, like for the wild type dynamics, neural stem cell counts, fractions. We can extend the model with feeder compartments. We have progenitor counts, we have neuroblast counts. Also, if we extend the model, we can really use this first scenario when we have this feedback from the downstream compartment. And black points are our data, and the 3 lines are different scenarios. And you see, except of very early times, so like, let's say we don't know initial conditions, so we know where we really start. All 3 models describe very similar dynamics. So again, if we were choosing our favorite scenario, we could say that we verified the hypothesis. But in fact, since we come with 3 hypotheses, then we cannot really distinguish. And this shows that, in fact, we need a different type of data to be able to distinguish, or we need this, what I was speaking about before, we need some more molecular information to be able to distinguish. So now we work on different big perturbation like chemotherapy applied to the cells and so on, but this is still a work in progress, and I would say too much chaos to speak about this. So then, before I would like to, as I said from the beginning, that I would like to talk about heterogeneity, but before speaking more about heterogeneity, I would like now a little bit to move to cancer and ask the question. So in the blood case, we saw that in fact all this stem cell derived dynamics is very essential for cancer.
Anna?
Yes.
Anna, here's Thomas. Yeah. Since this is a little end of a little section, shall we invite questions maybe?
Yeah, sure. Sure.
Yeah.
Yes.
So are there any questions for Anna on this part?
It was too simple, you know, small ODEs.
Small ODEs. I find this fascinating that with these little ODEs we can do so much. This is really amazing. Yeah.
Yeah.
Okay, I think there are no questions, so then you can continue.
Yeah, so now actually this was this question, can we learn anything from the models of cellular hierarchies in context of cancer? And again, in context of blood cancer, the answer was yes. And we in fact learned that self-renewal of cancer stem cells was the essential parameters for dynamics, but also for patient prognosis. So now here, as you can imagine, everything is even more tricky because our models are for mice. I would like to learn something for brain cancer, like glioblastoma. However, recent developments shows interesting aspects, and this is really work in progress. But I would like to show you this because, especially that you, Thomas, you have been working so much on glioblastoma. So I think maybe it's interesting also to see glioblastoma from this perspective of cellular hierarchies. So now here you see the distribution of different cell types in this single cell data. So again, just to remind you what I explained on Tuesday, that single cells are extracted, and for single cells, different transcripts are, level of different transcripts are measured. So this is transcriptomics, and this is highly dimensional set of data, but then using some, let's say, data analysis, mostly linear transformations, principal component analysis, and so on. The data, the information is reduced to lower dimension, and finally it's even here kind of projected in 2 dimension, but again, this is more illustration. But more or less what we see that that the shape of the data somehow reflects the cellular structures. You have some quiescent stem cells, then you have active stem cells, and then you have differentiated cells. So now, could we say about cellular hierarchies for data obtained from glioblastoma? So in this case, we work with tumors taken from patients. So that means data are coming really from clinics. And for different from different data, okay, so a lot of data we used so far were just from the literature, but it just started working also on the real data. That for such data kind of unexpected for me idea from experimentalists, how to, together with bioinformaticians, that you can assign this cellular type to the single cells, like if they are quiescence, active or differentiated, based on the transcriptomics profiles. So somehow you, for each cell, you know how different, which genes were expressed and how different, what is the, let's say, what are the features of the cells in this high dimensional space of transcripts. And then this was mapped to this information from healthy neurogenesis, where we know what are the, let's say, characteristics or transcript profiles of stem cells, quiescent stem cells, active or differentiated. And this how through this mapping, the cells from these tumors were assigned cellular type. So this gives us now this distribution of quiescence, active and differentiated cells for glioblastoma samples. Okay, so if we believe this, we may ask, do we learn anything from this? So on one side already here, you see that this is somehow better aligned. It seems better structures. It seems that they are better clustered. Here you already see that they are more mixed. I will show you also later other pictures showing that everything is now more heterogeneous or more chaotic. But what is interesting now, that looking now at the data from different samples, different tumors in different patients, you may look at this pattern, quiescent, orange, active. This you should sum up the dark gray and light gray because they are like two different types of active, but okay. For us, these are just active. And then green are differentiated. And you see that in fact, you have glioblastomas with a lot of quiescent stem cells, and or a lot of quiescent stem cells and actually no differentiated or nearly no differentiated. But you have also tumors with a little quiescent. And what you see here immediately is that, however, most tumor lineages are dominated by active stage cells. So it means cycling. Okay, although some of them are not really cycling, but they were already activated, just let's say taken out of this quiescent state. And then, okay, one question is it related to this transitions from quiescence to active? Or maybe is it related to transition from active to differentiated, to little differentiation? Or maybe it's again self-renewal, so maybe they do too much self-renewal and that's why you don't have so many differentiated cells. And in fact, the question is it important at all for dynamics? Because, I mean, what we care about is dynamics. And to connect this to dynamics, our colleagues in the lab, they did a statistical analysis. And so having information on the patient survival, and this analysis somehow indicates that having a lot of cues is good. Having a lot of cues is good, and having, so let's say, these are irrelevant. So somehow they hypothesize that transition from Q to A is the most important one. And transition from Q to A, it's again activation of the stem cell. So somehow it's again, we come back to the same parameter which we already before also found as an important. So now what can we do? Of course, if you think now about modeling of glioblastoma, you think about space, you think about the tumor structure, more diffuse, less diffuse, and so on, so on. But maybe first question is, could we learn anything just from a very simple model of cellular hierarchies? So could we then reformulate our model for healthy neurogenesis to describe these three types QAD from the model? And then, of course, we have now to think carefully what is really measured there. Quiescent cells are more or less this what were our quiescent cells before. Now, as I told you that as active cells, they put together cells which are activated, but also this early progenitors. And then as differentiated cells, they have more or less this what are neuroblasts in the healthy model. So somehow to describe it, we need to, the most optimal would be, of course, to collapse all these things to one equation. But this is pretty tricky because then we need some extra parameters. So maybe the easiest things, we did it also, but the easiest thing what we can do is just to say, okay, we can put all tabs together as one equation. And then we can have a model for quiescence, active, tabs, and differentiated. And then we can compare to the data quiescence to quiescence, our A plus T to their A, and differentiated to differentiated. And when we wrote model like this, it is at the first moment just linear ODE. So there are two issues. One thing is we don't consider regulation. Of course, we could now consider regulation, which we learned from the healthy, but the question is it really still regulated? And you know, the dynamics of cell population is significantly different than before, because in the healthy neurogenesis, you have this decline in time. While in the cancer, of course, we have growth. So in fact, this is clear that now self-renewal is large, must be large. And in fact, it is also clear that we again don't come to any steady state in cell numbers because we have a growth. And even what they did also with this data, with the cells which they got from patients, is that they managed to produce, now it's also a very initial stage, but they already managed to produce organoids from this. So they put it in vitro, and from this tumor cells, organoids are growing. So you see, even in vitro, that the things are growing. So the idea is that maybe as very first try to see if we can understand anything is just to ignore feedbacks, to say, okay, we have this initial stage when we have exponential growth of the tumor mass. And then in this first step, we also neglected space. And then you will see later maybe how this, which aspects of inclusion of space could then be important. If I have a linear model, then I have exponential growth, but we know that the distribution of different cell type converge pretty quickly to a steady state. Okay, you can see it in the simulations of our model, but this is also well understood, in fact, for much larger class of structure population models, not only for just linear ODE models. Like for ODE models, it's a consequence of Perron-Frobenius theorem, but you have also all these generalizations to infinitely dimensional models like structure population models, or using this Krein-Ruttmann theorem, or which in fact shows that you have convergence to the distribution, which is just given by this eigenfunction corresponding to the principal eigenvalue, and then there would be this exponential growth with the rate corresponding to this principal eigenvalue. So there was a lot of analytical work on this. I just put here two references, just quite new and quite like to one approach using this generalized relative entropy for structured population model by Petram, and the let's say more classical approach with Krein-Ruttmann theorem, and extensions and modifications are like done for even pretty generic structure, of course, under some assumptions, important assumptions, but for structured population model, starting from Glenn Webb, but also Pierre Magal and many others. So this is somehow what we know that we could generalize here also to the models which would be more complex. Also, I think we could include some spatial spread. And then what our initial experiments with organoids already showed, that if we take this sample and put in vitro and it grows, then it seems that the pattern, like the proportions, are also preserved, so that it's indeed a proportion of different cells makes sense. And this was essential question. Since this is what experimentalists used to conclude from their data, because the only what they use, they take data and they look what is the proportion of different cell types. So if it would be time dependent, then of course it would depend on the moment when you got the tumor out. So it would not give us any information. But now it seems that these proportions are meaningful, or maybe meaningful. So what we could do, we could, in fact, with the model, reproduce all this data. So here you have, I don't know if you see it, because so here, the let's say big bars are related to the data. So you have with orange, you see proportion of quiescent cells, then active cells, all active, and then this differentiated. And then in the middle, we put with a little bit darker color, simulated. So we estimated parameters for each of this distribution, and in fact, we could reproduce them. Each of them could be reproduced. So it shows that even that with such model, we have this ability to reproduce different proportions. What can we learn from this? Of course, I mean, what you see at the first glance is that that you may have different proportions, but then when we order them concerning time to diagnosis, then you see that in fact, so this is like fast growth, so quick detection, it means fast growth of the tumor versus slow. So you see that green cells may appear in a different way, but what you see is indeed that active cells are not active, yes, active. Oh, yes, correct. It's active cells behave in a pretty monotonous way. So it means if you have a lot of active cells, it is fast growth, so it's bad prognosis. So it also somehow corresponds already, that's why they also got it doing more careful statistical analysis. So what can we do with models? We would like to connect this to parameters now. So it means, but what I know through the model is that if I have my parameter estimation, I know which parameter choice I have for exactly this pattern. Okay, so now we can look at this. And if you look at time to diagnosis versus activation rate, because already before we have this hypothesis that's activation, which is essential, then you see that for small activation rate, there is nice monotonous behavior. So it means if it's very small activation rate, then time to diagnosis is large. While when activation rate grows, the time to diagnosis goes down very quickly. Then you see that if activation rate would be large, that time to diagnosis would be very short. However, for these intermediate values, in fact, it could be very different. And then we try to understand how the other parameters come into the play. And the only what you maybe can say here that for this activation rates intermediate, then self-renewal is important. These are these red dots. Then if you have intermediate activation rates, then if you take high self-renewal, that it will be time to diagnose. So high self-renewal is here. High self-renewal, then time to diagnosis will be short. Small self-renewal, smaller self-renewal, then time to diagnosis will be large. Anyhow, also here you see that in fact, of course, to have growth, I always estimate high self-renewal. So even from this model, you already can somehow see that it's activation rates which are essential for the, let's say, rate of growth and progression of the disease. And we have to be aware, of course, that we speak about glioblastoma. So, you know, even short versus time, okay, one can say it's important because this is like maybe 40 days versus 160 days. But we all know that in case of glioblastoma, anyhow, all this dynamics is very fast and it's not very good. So can we have any, let's say, conclusions from all this that are maybe promising in any sense, of course, except of the fact that we want to understand. So the idea is that since we see that activation rates is so important, then the idea is that maybe if we could make cells like reduce their activation, block them more in quiescence, it could help. And then another idea is that maybe if they are blocked to quiescence, maybe then if one activates them, one could get them all together from this quiescence and then maybe attack better with other therapy, with some therapy. So this are again very preliminary results, what I'm showing you. But here one idea was injecting one of these inhibitors of this Wnt signaling, which we already before identified as essential and connected to changes of the quiescence. And then, so this was now like this tumor cells which we got from patients, it was like it was like from one of our sample, like this sample, was somehow treated and then put into mouse, so like such a graft, and then also with injection of this Wnt inhibitor. And what was observed that indeed survival of this mice was significantly better than without this injection of this inhibitor. And we see also that this injection of the inhibitor changed the proportion of the, like increased the proportion of the quiescent stem cells and decrease the proportion of the active stem cells. And we see also that the proportion of the cells, in fact, this is the same as in this picture, that the proportion of the cells in quiescence in case of this this graft with injection shifted, like the proportion of quiescent cells is much larger, while proportion of active cells is smaller. So it somehow shows that this is maybe some promising thing, and this again points to this Wnt signaling. And again, I don't want to speak here about Wnt signaling because it's still, let's say, not really clear, and it's very chaotic and complicated at the moment. But somehow the models suggest some, or let's say, models help systematically thinking about this, what's going on, and about this, how we can maybe introduce some intervention. Okay, so now I just would like to tell you, so I don't know also, do you have any question for this cancer stuff? Okay, so I would then move to heterogeneity just to, so I always so far speak about just few compartments, and like all quiescent cells have the same activation rate, or all active cells have the same proliferation rate. So now is the question, can we understand? I mean, at the same time, we see all the single cell data which show great heterogeneity. So the question is this heterogeneity also important for the process? And to understand this a little bit better, we team up with a lab of François Guillemot in London. And they did such an experiment when they measured, like, again, they had mice of different age, and then they measure not only total and active neural stem cells, but also they measure neural stem cells which were cycling before and just stayed as stem cells. So somehow they get a marker during cycling which stays there. This way, we can distinguish between quiescent stem cells which were not activated yet, so it means they will be activated for the first time, from quiescent stem cells which were already cycling and come back to the stem cell pool. And so just distinguishing between these two populations, we put two equations. So these are only activated, and for this we'll have this transition back from active. And we can now estimate parameters. So of course, we can again estimate parameters for the total NSC counts and active NSCs. But the most important are this new type of cells which are measured, this resting neural stem cell counts. And if we would say that all quiescent cells have the same activation rate, so it means R1 would be equal to R2, we could never recover this dynamics. Then, in fact, we could hardly see any resting stem cells. While we see this resting stem cells like they are here growing. And to recover this dynamics, we estimate parameters which show that activation rate of stem cells which were cycling before is 10 to the 5, yes, 10 to the power 5 larger than of the dormant stem cells which were not cycling before. So this is a significant difference. And this is also in agreement with some other, like especially developments in the system, when one already has also suggestions that cells which were cycling before, they are easier to get activated, much, much easier. But this shows also that population of the quiescent stem cells is not homogeneous. And this is, let's say, proof for this without looking at single cell data. And now if we come to the single cell data, actually I explained it to you already on Tuesday, and today also referred to this several times. So we ask, can we get more information from this data than from all this other data? And now, of course, one could say, okay, now I have really a more detailed distribution. Maybe I could describe all of them through equations. But there are several challenges or several issues with the single cell data, which somehow detach them still from typical mechanistic modeling approaches. One thing is that usually we don't have time resolution. The data are obtained for a single time point, and the data show distribution of different cell types. And since one knows that quiescent cells will be activated, producing then later progenitors and so on, so one builds on this interpretation that the cells which are sitting here will be moving along such line. This is the so-called pseudo time. But this pseudo time has nothing to do with real time. Already before we learned that some of the cells after activation can come back, or this is only showing the, let's say, direction of a lineage and not real time. And what one tries to do with such data, there are more and more attempts to describe like flow of such different distributions using, for example, optimal transport. I don't know if you are familiar with optimal transport, but what it means at the end, it means that somehow this distributions along this pseudo time, which is just, let's say, constructed just from the data kind of average line. And to be able to find such line, you need this data distribution which is elongated. I mean, if it would be kind of not having direction, you could not even do it. And then along this data, you just take different distribution along this data, and then you implement this optimal transport, what means at the end that you just approximate this changes of these distributions along the pseudo time with linear transport process. Yes, so it's really like neglecting all growth and all other aspects of the process which we found above and below and before, and which also we know from experiments. Another funny issue is that if people put such U maps, you see some kind of these colors and you think, okay, here you have these cells, here you have these cells, here you have these cells. But if you really look at the markers of different cell types along this pseudotime, you see that that's not really clustered in the way as we would like to have it clustered. So it's not that if I have certain position along this pseudotime, I know uniquely which cell type is there. I can speak about some likelihood, or I would need to have some ability of having some overlapping distributions. So it also shows, in fact, that maybe this pseudotime is to reduce direction to understand the data. So, and then last but not least, from the challenges I would like to show you, and this I find, okay, now it moved. I found it, sorry. I found it pretty fascinating because, because before we always considered all these cell types in compartments. Yes, so one said, okay, we have quiescent cells, then they get activated, you have transition to another, you have transition to another, you have to transition to another. Now, if one sees these dense clouds of points, experimentalists started thinking it means processes are continuous. So it means that to get active, a cell needs to be transported through this area of quiescence. So it means cell which is sitting here will go, unfortunately now, so somehow the figure was moved. The blue line should go, of course, through this. So it's transported through different points in a continuous way here to get activated. And so on and so on. And it seems that it's not obvious at all from such data. I mean, these are only dense outcome of the pretty dense outcome of the process. One can also imagine that the cells can be activated in different points. So it's kind of discreteness or jump aspect. And then after division, they also can land in different points. So even questions, if one could distinguish between just such hypothetical scenario, I would say it's a relevant question for modeling. Just these pictures so far are just pictures and visualization and not really, it is really sometimes difficult to conclude about processes from them. So then in the last maybe 10 minutes, I would like to tell you some ideas, like mathematical ideas, of a framework which could allow maybe accounting for this complexity of single cell data. So somehow which problems we would like to solve with this. One thing is, as I showed you, that I have multiple scales of distributions. So it is not that I can divide this data into pieces, count them together and put to my compartmental model, because we tried this. This does not work. You don't have any reasonable match. So maybe one should look at these distributions and as we present them here, or it's in some way. Another thing is that we may have coexistence of continuous pieces and discrete. Like here, if you think maybe it's indeed maturation process is a continuous process, but then activation is kind of a point where the cells just get cycling. And also, it's very often if people work on this data that they already reduce them to some Riemannian manifolds or some graphs. So somehow the question is in which state spaces we should work. Another problems with all these stem cell based models, as we also discussed on Tuesday, is that you may have selection, like that the cell types, this is like a picture from the Tuesday, that in time, a continuous distribution of cells somehow concentrated in single point. Or you may have also situations maybe when you want to start from cells which are the same, like homogeneous, so you have concentration in such a space. So all this suggests that maybe we should think about a model which is operating in some, first of all, on a pretty generic state space, and secondly, describing cell distribution in terms of measures. Actually, it would be nice if we could speak about probability measures, but since we have growth in the system, we don't have conservation of total mass, so it's not really probability. But it's like in our glioblastoma case that you may have these distributions, and then let's say growth or decline of the total mass. And this, let's say, if you want to write an equation formally, then it's, of course, pretty easy. You just say, okay, we take classical structure population model. This is the most generic. So it means you have time evolution is governed by transport. So it would be like this transport through quiescence, for example, where you need some velocity. Then you have some growth or decline. And then you have also some non-local operators. This could be like also as mutations on on Tuesday, but in general, it could be also a like kind of distribution after division, which is not necessarily concentrated or things like this. And then is the question, can we work with such models? And just I would like, I don't want to go here into details, but I would like to tell you that in fact, putting a mathematically consistent framework for such equations was also interesting mathematically. So, okay, so this is exactly what we want to do. Now, instead of thinking about movement of single cells, we want to think about distributions and evolution of this distribution. And then we can consider it, in this I will show you, we can consider it in pretty generic spaces of measures, like radon measures, which is just finite and assigned. And for this, you need, of course, a good notion of the distance. And since, what would be typical notion of distance for problems in measures? So it would be total variation. But total variation...
Can I ask you?
Yes.
It looks to me like you don't say explicitly, but you are now in the world of optimal transport, because you are transporting, you are moving between measures, you talk in the language of measures, and if you ask which distance, maybe the Wasserstein may be useful.
Yeah, no, it's a very good point. And in fact, I wanted to come to this just to speak about this maybe here. If you think about optimal, what is optimal transport? Optimal transport is just transport of these distributions. So first of all, as you also pointed to, saying Wasserstein. Why Wasserstein? Because in optimal transport, you have just transport of this mass, like you can change distribution, but you have no growth, no decline. That's why you can use Wasserstein, which is good for measures with conserved total mass. So if we would, from our model, remove right-hand side, or maybe even just remove this growth term and just leave some only transport, it would be exactly like PDE for optimal, corresponding to optimal transport. In our case, we have growth because we speak all the time about cell divisions. So it's clear, like, as I also showed you in this simple models, in case of glioblastoma, you have growth. In case of healthy situation, you have decline. So we cannot use just transport. And we had, that's why we have to, it's like Wasserstein, we have to come with kind of modification of the Wasserstein distance. Maybe what is also here important from my perspective, this is that it's not that I only want to, let's say, describe evolution, like mimic evolution and find how individual maybe cells moved. I want to understand how this regulation of this process is the same what we did for ODEs. We would like also now to understand which processes change in aging, which processes change in cancer, which processes change after perturbation. So somehow I still would like to have, let's say, process-based model rather than data-based description of the flow as in optimal transport. I don't know if it's
No,
clear.
that helps a lot, but I have an idea that may be useful or not.
Yes.
Could it, some of the difficulties you are facing here have to do with the fact that we are going binary on between homogeneous and heterogeneity, and maybe partial heterogeneity, to introduce it, may help you to kind of, in quotes, cluster the distributions that are possible here.
Yes, I mean,
It's an idea. I'm not sure it helps
yeah,
here
yeah, no. I mean,
or relevant.
this is exactly what in fact, this is what I thought when I saw this data for the first time. It is that you can cluster them as they cluster them with markers. The problem is that these markers, they don't at all contribute to this what we had before. So somehow, I think it can be better maybe understood with this picture. Because if people plot such, visualize this data in this way, you have a feeling that you have here really clusters. But here, in fact, you have these different data sets along this pseudo time, they overlap. So, okay, maybe I should say it differently. It's like, I wanted now to show you different challenges related to this data. And I don't have a good models which take into account all these aspects. And my conclusion is that, in fact, we should do different things depending on the questions. So sometimes I think, yes, we should aggregate them and put them together as clusters. One of the question when I would like to have this continuous transition is exactly this question. Is the process continuous? Because again, for me, the fact that I have dense cloud of points does not mean that the cells which is sitting here will move in a continuous way through this cloud. It could be also that this cells is activated just with a different activation rate than the cells which is maybe further this direction. And what I would like...
About the biology, I believe that things are not continuous.
Okay.
Because there is a trigger, there is a threshold, there is systems that have to reach certain value before something happens or doesn't.
But it would be nice if you, but because I tell you, my biologists at the moment here at the Cancer Research Center, they believe that it's continuous. And in fact, I believe that it's not necessarily continuous. And I would like to show them this, showing that you may have different models producing exactly the same data. So somehow that you cannot decide based on this data. I remember when we introduced this first continuous structured population models for blood 10 years ago, then indeed, people at that time were all convinced processes are not continuous. And now I think that many biologists are really misled by this data and started believing that you have here continuous transitions. So I like it that you say that it's not continuous, because this is what I think, at least I think, that you cannot conclude from such data about continuity of the process.
I would go back to the definition of pseudo-time.
Mm-hmm.
Because this is first time I hear it, but it's kind of, you scale the time in different way, or you do the snapshots not in 1 minute. I'm not clear about that.
Yeah, no, it's even worse. You don't have here any real times or snapshots where I have this pseudo time again. Okay, here. This is just kind of average line, average trajectory through the data. The story is like this. If you have this data and you know that these are stem cells and these are active and these are neuroblasts and these are neurons, then since biologists know that these guys will produce these guys and these guys will produce these guys and these guys will produce these guys, then they say, okay, it means that this line reflects time. But it's not, the data are from single point time point measurement. So you don't have at all, you don't see at all how single points were transferred or transported or moved in time. this is in fact one of the biggest challenges. Again, the field is now very dynamical, so maybe I don't know about different systems, but for example,
but the trouble could be, sorry, the trouble could be that while averaging may ease some calculations or processes or graphs, some misleading element in averaging. And maybe that's a difficulty that you are stumbling on to interpret the picture. It's because it's averaging.
Yeah, this also, but also, first of all, I think one of the most important thing is to get data with time resolution, and this is what they now promise. They come with a new concept. They call it time machine, when they will have values for 2 different time points. Even the question is where daughter cells. So the cells here divide, and when the daughter cell is in the same place and then moves away, or it is after division already somewhere else. All these questions, at least in context of neurogenesis, are really not answered. And in fact, I would say at this level that the main role of mathematical modeling is that we pose systematic questions, and then they see how unsystematic is the whole reasoning about these pictures, because there is a lot of conclusion by biologists are drawn based on the pictures, and this is not. That's why actually, if we ask ourselves what we could do now mathematically, I thought we should build toy models, prototype models with different assumptions, and showing them that in fact, I believe you probably cannot distinguish between using such data, and then design experiments which would let us getting better data.
And these difficulties make it fascinating, but
Yeah.
I have some questions. I don't know if you can handle questions now, but
Sure. I mean, I can.
I have a sequence of questions.
Yeah. So
one is on one slide you had comparison of.
Yeah, Itamar, sorry to interrupt. Why don't we let Anna finish first, and then we do the question?
Of course, yeah, you're right.
Yeah, thank you. Okay.
Okay, so I maybe then I will shorten this part. I just wanted to tell you that this was like, a little bit perpendicular story, that in fact, thinking about these distributions, we thought, can we describe PDEs in terms of measure evolution? But then of course you need a good metric, and that's how it was now already suggested. It should be something a little bit related to Wasserstein, but also allowing for the growth. And a good candidate, it's so-called flat norm, or Kantorowicz-Rubinstein norm, or bounded Lipschitz norm, which can be understood in this form of duality. So if you compare it now to Wasserstein, would be that I need now bounded Lipschitz functions as test functions, which have also Lipschitz constant bounded. So somehow we can find a good metric which allows you that if you have 2 Diracs and you make them closer, that the distance between them is also converging to 0. So it is like in Wasserstein, exactly like in Wasserstein. The only point is that for me, I may have 2 Diracs with different masses. So somehow now the optimal transport is on one side putting them together as in Wasserstein, and then making their mass, let's say, equal. So this is just for intuition. And then for these spaces, we could, like for the measures with this metric, we could really now, let's say, prove that the space has a good structure, that positive cone is even complete, and then so we can apply Banach fixed point theorem, and so that we have a good functional analytic setting for studying these models. And first, what we did for these models, I mean, this I can skip, is that we showed for these models Wells, Posnet, and Lipschitz continuity with respect to model ingredients. And now we can, let's say, this is a very generic framework. In fact, in this framework, you could put also this famous fragmentation coagulation models, or you could put models when velocity is not just this is just also a distribution of velocities. Like you may have here a lot of generalizations. And one of the nice generalization is that this model does not need to be on Rn. You can also think that these things are happening on graphs. And in fact, you have some indication for graphs if you apply topological data analysis, when you just then maybe see that between some groups of cells, there are different connections than in the other groups of cells. Again, this is just data analysis and visualization. So again, from this alone, we don't learn about processes. So we may also put this data in hyperbolic, look at this data in different geometry, like using Poincaré maps. And you see that you always see kind of the trend of moving one cell type to another, but somehow it might be that from this kind of more, let's say, sophisticated mathematical data analysis, we can get more hints. But this is all of this are now open problems. Actually, since I was too long with time, maybe I should skip the rest now. Yes, no. So maybe I just summarize it. So nice, and then we come to the questions. Maybe then through the questions we come back to more mathematical aspect. So just I wanted to show that, like, with this development of mathematical framework, just depending on the type of data and type of questions, we see that stem cell-based systems exhibit heterogeneity at different levels. And this functional relevance of this heterogeneity is not well understood. So if something is very heterogeneous, it does not yet mean that this heterogeneity is important for process. And this is what is new when you want to bring mathematical modeling or mechanistic modeling. So our differential equations to the business is that we have to connect them to this data analysis problems. Then, of course, as I said, let's say my main effort last year was to understand what bioinformaticians are doing there, to understand what are really questions or problems. But now is the question how we can, let's say, use this data and what we can say about the processes using our mechanistic models. And with this, I would like to finish and just to put for the today part, this was this neural stem cell models, which were developed together with my PhD student, Thomas Stiel, Joa Holly, and Diana Patricia Danchu, and with this close collaboration with the group at German Cancer Research Center of a group of Anna Martin Villalba, when the main experiments presented today were done by Georgios Kalamakis. And I also showed you this heterogeneity of quiescent stem cells done together with the lab of Francois Guillemot, and the analytical framework we developed now with Piotr Gwiazda. So this was kind of, let's say, linking just purely mathematical considerations to this. Yes, thank you.
Yeah, thank you very much, Anna. This was wonderful. Thank you for having the time to talk to us and giving these beautiful lectures. So let's open the floor.